Welcome to section 9.1, the structure of DNA. In this section, we'll explore how DNA is built, from its molecular components to its iconic double helix shape. We'll also look at how DNA is packaged inside both prokaryotic and eukaryotic cells, and why that packaging matters. The section lays the groundwork for understanding how genetic information is stored and passed on in all living things. These are our goals for the section. By the end, you should be able to describe DNA structure and explain how DNA is arranged in both prokaryotic and eukaryotic cells. DNA's double helix didn't come from just one discovery. It was a puzzle solved by multiple scientists. Rosalind Franklin's X-ray image, photo 51, gave a critical clue about the helix shape. Maurice Wilkins, her colleague, also worked with X-ray diffraction. Watson and Crick then used insights from Franklin's data to build their famous model on February 28, 1953. In 1962, Watson, Crick, and Wilkins received the Nobel Prize, but Franklin had passed away in 1958, so her essential contribution wasn't honored in the same way. DNA is made of repeating units called nucleotides. Each nucleotide has three parts: a sugar, a phosphate group, and a nitrogenous base. You can think of these like Lego pieces that can snap together to build a much larger structure. Nitrogenous bases come in two families. Pyrimidines are single-ringed structures, cytosine and thymine, and in RNA, there's uracil. Purines are larger, double-ringed structures, adenine and guanine. A helpful trick to remember these: cut the pie. Cytosine, uracil, thymine. Those are the pyrimidines. The differences in size are important for base pairing. DNA forms a right-handed double helix. You can picture a twisted ladder. The two strands run in opposite directions, which we call antiparallel. The rungs are the base pairs. A pairs with T through two hydrogen bonds, and C pairs with G through three hydrogen bonds. These pairings keep the helix stable and ensure faithful copying of genetic information. When we say DNA is right-handed, we mean the helix twists upward to the right, like a standard spiral staircase. A left-handed helix would twist the other way. DNA's right-handedness is a universal property of life as we know it. Erwin Chargaff made two key observations. First, base pairing is consistent. A always equals T, and C always equals G. Second, the ratios hold true across all DNA, giving structural balance. These rules help explain why the double helix is so stable. It's like every puzzle piece has only one correct match. In prokaryotes, like bacteria, DNA is a single circular chromosome located in the nucleoid region of the cytoplasm. To fit inside the cell, it's supercoiled, twisted and folded tightly. Imagine trying to stuff a long rope into a backpack. Coiling it up makes it manageable. Eukaryotic DNA is more complex. It's stored in a membrane-bound nucleus, and the chromosomes are linear, not circular. DNA wraps around histone proteins to form nucleosomes, often described as beads on a string. These beads coil further to form condensed chromosomes, especially visible during mitosis. Not all DNA is packaged the same way. Heterochromatin is tightly packed, and its genes are usually inactive. Euchromatin is loosely packed, leaving its genes accessible and active. This organization helps the cell regulate which genes are turned on or off. DNA structure, the double helix with complementary base pairing, explains how it can store and copy genetic information. Prokaryotic and eukaryotic cells package DNA differently, but both strategies solve the same problem: how to fit a huge amount of information into a tiny cell. Understanding this structure is the foundation for all of genetics.